So this is a mini lecture about genome sequencing using for newborn screening. And usually I start off with a second picture, the second slide's always a picture. Kind of this is an overview, bird's eye view of the topic. And we have 3 learning objectives. They're all kind of related. The first is to describe the process of genome sequencing for babies. And then part of that, there are logistics to how it's done, but there's also various methods and various levels of resolution. So there are 5 commonly used methods at very different levels of resolution. So the second learning objective is to be able to summarize those. And the third learning objective is one particular problem, kind of focusing now a bit deeper, and one common problem that you can actually see at the whole genome level. And this has to do with disorders of sexual development, and we'll talk more about that. But that's the 3 learning objectives. And this little mini lecture came about as this is from my inbox, an email, and about an article that came out about genome screening. And so that's kind of behind this. And there are some questions with this. The first question is, what is meant by genomics? I think a lot of people use the word and don't fully understand what it is. So what is meant by genomics? How does one do genomic screening of newborns? Why should we screen everybody, that is at the population level? What are the different levels of genome sequencing strategies? And we'll talk more about that, but there's, as in the learning objectives, there's 5 different levels. And then finally, how frequent are disorders of sexual development? So what is meant by genome? And you can look this up. There's also a link here to the wiki page. You can look this up in the Oxford English Dictionary. It's pretty simple. It's the haploid set of chromosomes. So humans, we have 23 chromosomes in sperm and the egg. All the other cells have 2 copies, so we have 46 chromosomes. And so the genome is simply, your 1 copy of the chromosomes, all the genes, or more recently, all the DNA. Ome means all. And that's very important because a lot of people confuse genetics and genomics. A gene could code for a protein, or it could also just code for a stable RNA. But so there are lots of genes in the human genome, but the genome is all the genes, all the DNA. So in the Oxford English Dictionary, it gives examples when the word is used. And one of the interesting things, 1970, there's an article that says human genome contains perhaps 10 million genes. It was in the 70s. In the 80s, they thought, okay, maybe it's 1 million genes. In the 90s, they thought it was 100,000 genes. And then early 2000s, they actually had a draft of the human genome. They kind of finished a draft, which is about 85–90% done, but there's lots and lots of gaps. And at that time, they said, oh, there's only 20,000 genes, but the gene cloning methods are really poor. You have lots of genes that are alternatively spliced. So that number, yeah. And now we know that lots and lots, in fact, there are many more genes that code RNA, and don't get translated into protein. So there probably are at least 100,000 genes, certainly different products that are transcribed. Okay, so here's the article: genome screening and genome sequencing and newborn screening, balancing consent with the right of asymptomatic at-risk child we found. And so the idea is that, yes, they need to consent to this, but by screening everybody, particularly for rare diseases, sometimes you can find treatable genetic problems that could actually, that would be found by screening everybody. So that's the kind of the gist behind this. So in this paper, they look at, this is done by a group in the UK, and they look at what is actually being sequenced, and they look at a variety of different studies, some in Australia, in the United States, in Greece, and the UK. And most of these studies, they're doing a few hundred genes, 600, 400, 180, 500, 223 individual conditions caused by 500 genes. So this sounds like a lot, but again, remember, genome, ome means all. They're looking at a few genes, and these assays, sometimes some of them are a little bit more accurate than others. And so one of the questions is, how good is these tests? How reliable are they? And this is an overview looking at the different types of things that they could screen for and how well they work. And you can see there's one particular test for osteogenesis imperfecta that's 100% accurate. If it says positive, then you know that the child has this disease. And there's a link to it. I put a PDF link to the paper in there. So that case works 100%. But if you look at all these others, organogenesis less than 12%, autism spectrum disorder. These tests, they're accurate maybe 8% of the time. That's not real good. So a lot of these, cardiovascular, some of these congenital heart disease is correct 9.7% of the time. I mean, maybe we're having to look, but these numbers are pretty low, the bottom line. And so one has to kind of understand a bit the value for these. Okay, so and this is kind of an overview. I like this picture showing the relationship between the laboratory and the clinician. And so basically the genome sequencing is done in the lab, and they extract from the baby's foot or wherever a cord sample or something, and take some blood. You sequence it, and then we look for these different parameters. Now, so the green is kind of the laboratory, and this is my background. I'm more a lab person. And the blue is the clinicians, the doctors. And the doctors, they look for clinical observations, things, floppy, jaundice, congenital heart disease. There are some things that can be looked for at the clinical level. You also put in factors, age, sex, family history, and you wind up with a set of terms. And so in the end, it's a combination both of the clinician and the laboratory results that decide what to do. And in some cases, most of the time children are healthy, so no further actions needed. Sometimes you need to refer to clinical genetics to look more into it. And there are times where you need to kind of manage it as appropriate. So that's kind of the overview. Like I said, I like this article for showing things, what's going on. Getting back to this article that came out in my email, in my inbox, basically looking at the population level for newborn is not primarily about individuals, parents, or families, although of course they should be engaged, but rather it's finding the at-risk child. And so the idea is that if we screen, we can find some of these rare things. That's the gist of this article. So why screen for everyone? At the population level, then you can find some of these at-risk patients. So I said before that there's 5 different levels, and this is kind of a graphic of it here. And the highest levels of chromosome, and this they can do for 100 years. So you can take pictures of chromosome, and 50 years ago, genetics class was doing this kind of thing. When you do a squash and you can look and see the chromosomes. You can do something called G banding where you can see this. And now, so the resolution is it is the genome, everything, but you can only see it in a microscope, so you just see these lines. Is the chromosome there? Yes or no? But it turns out that's an important question. We'll come back to that in a minute. And you can do G bands, and that can give you resolution. So an entire chromosome could be 100 million bases long. The G band can resolve things in the order of 5 to 10 million bases. Microarrays, which are little small pieces of DNA, 5 to 100 kilobase regions, which is great, but I should point out that this is a lot more noisy than people think, because almost everybody you talk to somehow manages to forget that the human genome is full of repeats. Like the allele repeat is repeated 500,000 times in the human genome. And so the 300 base repeats. There's lots and lots and lots of repeats. 2/3 of the human genome is repeats. And so these microarrays are a lot more noisy than people think. And that's something to be considered of as well. But anyway, you can get resolution, in the range from, supposedly 50 bases all the way to 2 million. But there's a lot of caveats with that. Then you can actually sequence. And now we have the genome sequence, but again, the technology, we haven't finished the human genome yet. It's getting close. We started it 40 years ago, the Human Genome Project, but there's been some problems. But so presumably you can get down to 1 base resolution. There's something called whole exome, and these are when you make the genes in human genome encode proteins, but they're spliced. And so only about 1% of the human genome actually encodes proteins, but that 1% has lots of spaces in between where things get spliced out. And so what you can do, it saves a lot of money if you can only sequence, you know, basically 1% of the human genome is a lot cheaper than doing 100%, right? It's literally 100 times cheaper. But you're missing a lot. And so that's the whole exome sequencing, as opposed to whole genome sequencing, where you can do from beginning to end, from telomere to telomere, and they're starting to do that now. But this is kind of, they haven't done it yet. So anyway, so this is kind of the range, and at the very bottom, the diagnostic yield goes from low to high, and incidental findings low to high. Variance per person, you know, at the chromosome level, either the chromosome's there or not. G band, similar to just looking, is it there? Microarrays, you could get hundreds of differences. Whole exomes around 20,000, because I think there's 20,000 genes, but like I said, that number should be treated with very, very, very careful. But you know, roughly 20,000. Whereas you have the sequence, you have 4 to 5 million differences, but to be honest, it's probably going to be closer to billions when they do this. And there's more than 2 billion SNPs out of the 3 billion base pair. So yeah, you're going to see lots of differences. In fact, even within the same person, every time your cells divide, there's thousands of differences. So you're going to see lots and lots of differences. Okay, so that's where that is. And the G bands. Now, we were talking earlier about banding, and this is an example on the upper left corner of banding of chromosomes. And what we see is that you can see all 23 chromosomes and an X and a Y. So this is a male, presumably. And when I taught a genetics class, and the lab technician told me that roughly 1 out of every 100 people, the sex chromosomes don't line up. That kind of been his experience. And sure enough, we had lots of students in there, and they would do the drop blood their own karyotype, and there were people in there that were clearly male, and they had 2 X chromosomes, and people that were clearly female had a Y chromosome. When this happens fairly often, and this is kind of what got me interested in the next part. So just to finish this, on the upper left is the kind of the staining of the chromosomes, which they can do that for many years, but more recently now they can actually color code each chromosome, and you can see all the different ones, and you can see sometimes rearrangements. And this happens actually frequently, and then even within the same person, like I said, there's variation every time your cells divide. Okay, so getting back to this idea that 1 out of every 100 persons has this, what it's called, differences or disorders in sex development. This is a pretty astounding number, and I find it kind of interesting, but also in terms of screening for newborns, this would be useful because, so for example, you know, imagine, and they have examples in this article about, you know, people that are, you know, as adults discover that they have different chromosomes than they thought, and so that would be useful in treating babies sometimes, just knowing what, you know, because sometimes you have, for example, 3 copies of the X chromosome or 3 copies of chromosome 21. That's Down syndrome. That's easily diagnosable. But there's also sometimes it's useful to know, make sure you have the right number of chromosomes. And actually, surprisingly, it's not nearly as efficient as you would think. Only 1 out of 3 pregnancies is successful, and most of the time the reason it doesn't work is there's not the right number of chromosomes. There's a variety of things for that. But if you look at the sex spectrum, so the typical male, and by typical is 99% males, but not 100%, has an X and a Y. And then similarly, the typical female has 2 X's, but not all, 99%. And so this kind of gives an overview looking at this. So finally, we're back to our questions, and I'll put some links here for you to look at. What is meant by genomics? There's a lecture on the Human Genome Project. How does one do genomic screening of newborns? There's a review article that talks about this. Why screen everybody? This is the original article that I talked about at the beginning. What are different levels? And again, there's this article that I provided earlier talked about this. You go to PubMed and type in genomic screening babies, you're going to get more than 14,000 articles. And so there's, let's see, you can kind of poke around and maybe look. How frequent are disorders of sexual development? Well, there's a link to the Wikipedia page and also a PDF article about this. And that's it. Thank you very much.